typical question you should expect is to be able to understand the growth curve, which is a plot of growth against time. And if you look at this curve, it has what you call the sigmoidal shape, which is the S shape. And at this early phase here, you see there is a little bit of growth, where it's a delayed growth, and this phase is called the lag phase, where the cells are building up nutrient, taking a lot of factors in before they begin to grow. But after they overcome this lag phase, the cells or the organism experience an exponential phase, which is otherwise called the log phase, and that is this portion of the curve. And at this point, the growth then gets to a climax, and then it begins to diminish. So you have diminished growth at this point, and after that, the cells become stationary. So you have this four component of the growth curve: the lag phase, the log phase, the diminishing phase, and the stationary phase. So you can find all these details in your biology textbook, and then experiment on growth, and finally we're moving on to talk about question. So we'll be using this question and answer to further teach on the topic. So let's begin with the first question. Our first question is the irreversible life process by which new protoplasm is added to increase the size and weight of an organism can be termed as anabolism, catabolism, growth, development. This is a no-brainer. So the answer to this is growth, which is an irreversible increase in size and weight of an organism by the addition of protoplasm. I'm going to get to that. The next question says biological growth refers strictly to an increase in the right answer to this question. If you consider all the options here, the right answer is protoplasm of an organism. Now I'm going to quickly explain what that means. Of course, the term protoplasm, if you just look it up online, it simply means the living part of the cell that is surrounded by a plasma membrane. So it's a jelly-like, colorless, transparent, and viscous substance. That's what the protoplasm is about. And it contains various cellular organelles and molecules. So protoplasm is responsible for various living processes and is sometimes divided into cytoplasm and nucleoplasm. So that's a component of the cell and component of the nucleus. So the term protoplasm, however, is not often used in modern biology. You won't see that in modern biology textbook because it's based on a simplistic view of the cell structure. Now we know the cell to be very complex in nature, and so you will not just treat cells as one jelly-like, colorless, transparent, viscous substance anymore. So this is an old question. I don't see JAMB repeating this kind of question anymore. But don't be surprised. This question was asked some 20 years ago, 1999. Okay, so the next question is from 1998. The most reliable estimate of growth is measured by changes in length, volume, surface area, dry weight. The right answer to this question is the use of dry weight. And why is that so? Changes in dry weight gives a reliable estimate of growth because by that you would have removed the water, and then you can then base your measurement on only the cells. Question number 4. For growth to occur in organisms, the rate of food storage must be low. B. Anabolism must exceed that of catabolism. And C. Catabolism must exceed that of anabolism. And D. Food storage must be high. The right answer to this question is option B. Growth occurs when anabolism, which is the buildup of complex molecules, exceed catabolism, which is the breakdown of complex molecules. Question number 5. A unicellular organism is said to have attained its maximum growth when it has reproduced itself several times, has used up all nutrients in the medium, can no longer reproduce itself, or D, can reproduce itself. Optimum growth is attained when organisms can reproduce themselves. When they get to that point where they can also produce other organisms, then their growth can be said to be maximum. Then question number 6. Growth in living things is brought about by deposition of new material from the surrounding, synthesis of new material from the surrounding, from within the body. C, absorption of water and minerals. D, elasticity of cell. The right answer to this is the synthesis of new material from within the body. Now let's look at growth in plants and animals. The next question here is from 1988, long time ago. The radicle of a bean seedling grows rapidly in the region of A, of the root tip, just around, B, just above the root tip, C, just around the root tip, and D, just below the root tip, just below the root tip. The right answer to this question 7 is um, um, B, just above the root tip. Of course, the radicle is develops into the root, so it's a miniature root. A little above the root tip is the actively dividing meristematic cells, and you know that these meristematic cells is what produce new growing cells. So the outermost layer of which forms the root cap and the piliferous layer. So you can look up that diagram of the internal structure of the root. We have covered that in previous topics. Question number 8. A seedling grown in dark is likely to be A, etiolated, B, dormant, C, just around, sorry, C, sturdy, and D, stunted. So the right answer to this question is etiolated. And what does that mean? Etiolation occurs when plant grows in the dark. It results in lengthening of internodes, reduced and pale yellow leaves due to absence of chlorophyll. So that is what you get when plant is grown in the dark. Now let's look at this question number 9. The area, of course, this is a growth curve, right? The area with the highest growth rate is labeled at what point do you have the area with the highest growth rate? Now, if you consider this diagram carefully, in this curve, you find that, of course, this is the starting point of growth, and this entire part, this 1 to 2, gives us, remember, what we call the lag phase. 1 to 2 gives us the lag phase. But from 2 to 3, this entire length here is the area of exponential growth, or the log phase. And therefore, Roman figure 3 is the highest growth rate, Roman figure 3. 4 here is the stationary phase. And what you have at 5 is the point of decline in growth. So the answer to that is option C. Question number 10. Environmental resistance is greatest at what point of this curve? Environmental resistance is at the point with the little growth. And which point gives us that little growth here? And that is this point that you have, which you have, which is the stationary point. That's where, hold on. Okay, environmental resistance will be, yeah, when there is no point, there is no more growth. So that will be Roman figure 4. Now, if you look at question 11 says, when the tip of coleoptile is illuminated from one side, it comes towards that side because growth hormone is dash A, concentrated on that side at the tip of the coleoptile, concentrated on the dark side at the tip of the coleoptile. C, destroyed by the light rays at the tip of the coleoptile. And D, concentrated below the region of the coleoptile. Okay, so if you want to take a guess at this question, what would your answer rather be? So usually what happens is that when you illuminate the plant from one direction, auxin, which is a growth hormone, would be unevenly distributed. So unilateral illumination of shoot causes auxin to move away from the light source. So when auxin moves away from the light source, it gives a greater concentration of auxin on the shaded side of the shoot, which in turn causes increased cell elongation. And as a result of that, that part where auxin is now concentrated, the dark side, will bend the shoot. So auxin becomes concentrated on the dark side of the tip of the coleoptile, which is option B. Question number 12. Growth in animals differs from that in plant because one occurs in the reproductive cells only, 2 or B, equally in all parts of the body. C is restricted in restricted part of the body. And D at the apex only. So, in other words, growth in plants, what makes growth in animals rather differ from growth in plants. So let's look at the options again. Growth in animal occurs in reproductive cells only. B, equally in all parts of the body. C, in restricted part of the body. And D, at the apex only. Okay, so the right answer to this question is B. Growth in animal is equal in all parts of the body. So that's what you call intercalary growth. And it's limited to certain period in life. So growth in animal doesn't occur throughout life, but it's equal in all parts, right? Whereas in plants, growth in plant is restricted to certain growing regions, particularly the apical meristems, and it occurs throughout life. So the right option would be option B. Let's consider question 13. Which of the following is the correct sequence of growth in multicellular organisms? So usually for growth to occur, you have cell division. The cells need to divide, they need to enlarge, and then they begin to differentiate. It is in this order: cell division, cell enlargement, and cell differentiation. Now let's move on to germination of seeds. Which question number 14? Which of the labeled parts will develop into a new maize plant? Now, this is the structure of a maize seed. Question number 14. So, which of these label parts will develop into a new maize plant? First and foremost, we need to understand what are these label parts. Number one is the fused pericarp and the tester. This is what forms the seed coat. So, number one is seed coat. Number two is um, the endosperm. Endosperm is what provides nutrients or nourish the embryo that is growing. And then number three is the growing embryo here, where you have the tip of it is what becomes the epicotyle, and the lower part of it becomes the hypocotyle. And then you have number four. Number four is called the scutellum, and then number five is the point of attachment to the cob. Okay, so I haven't labeled, understand what all these parts are. So which of the label parts will develop into a new maize plant? Of course, that will be number three, which is the embryo. Question number 15. The structured labeled one is I mentioned that already, that the structured labeled one is the seed coat, which is the fusion of pericarp and the tester. Now, question number 16. Question number 16. The main function of the structure labeled number two. What is the structure labeled number two? That is the the endosperm. The endosperm is the part labeled number two. What is the function of the endosperm? It nourishes the embryo and the growing part. So the right answer here is option C. Now let's go on to consider question number 17. It says dormancy in seeds may be caused by um, the following. Either any of the which of the following will result into dormancy in seed? The rapid diffusion of oxygen into the seed, delayed development of the embryo, the rapid absorption of water, and then the insufficient stored food for the embryo. Of course, a dormant seed is a viable but dry seed that can remain for a long period without germinating. So, dormancy ensures germination during a favorable season. So, seed dormancy may be reduced by growth inhibitors or the impermeability of the tester, that's the seed coat, and also delayed development and concentration of seed. So, which of this option looks or sound like what I've mentioned? And that would be option B, delayed development of the embryo. That is dormancy. Now let's look at types of germination. There are two types of germination. You have what you call the epigynous germination and the hypogynous germination. Let's look at the definition quickly. So in epigynous germination, the hypocotyle is rapidly So first and foremost, you need to understand what the hypocotyl is and what the epicotyl is. And for us to do that, I'll show you the diagram of the seed of a monocot and the seed of a dicot. Now, if you look at this diagram of a monocot, you see that you have the endosperm, as I mentioned earlier. You have the now. What I want us to focus on would be on this the embryo here. You can see the epicotyl and you can see the hypocotyl here. Similarly, if you consider a dicot seed, all right, you will also find that you have the epicotyl here and the hypocotyl just here. So what you have is the epicotyl is above, while the hypocotyl is below in relation to the cotyledon itself. So when germination occurs, the movement of the rate of movement of the epicotyl or the hypocotyl determines the type of germination that you get. Okay, so let me go back to the note. Now, in epigynous germination, I've understood what hypocotyl and epicotyl are. When you have the hypocotyl, which is the part that is growing towards the root, right? If this part rapidly elongates, it pulls the cotyledons and the plumule above the ground, like when you're growing your bean or your cowpea. So this is what you get in a dicot germination. Again, this is what a dicot looks like. So in this kind of germination, now the cotyledon is above the ground. Why? Because of the rapid movement, rapid growth of this hypocotyl. So because that happens so fast, the root is established on time, and then this has no choice. The upper part, the cotyledon, goes above the ground. While in the other case, hypogynous germination, the epicotyl is rapidly pushing up the plumule above the ground and leaving the cotyledons on the ground. And that is common in most monocots. So let's look at that. In monocot, you have, as a result of the rapid growth of the hypocotyl, sorry, as a result of the rapid growth, if you consider the growth of this hypocotyl and epicotyl, you see that the epicotyl is growing rapidly. So it leaves the cotyledon behind in the ground. Okay, so now that we've established the epicotyl and the hypocotyl and the cotyledon's position in these two types of germination, let us consider questions on types of germination. So in epigynous germination, it is the epicotyl that elongate fast, hypocotyl elongate fast, hypogynous that elongate fast, plumule that elongate fast, roots that elongate fast. Which of these options best describe epigynous germination? So in epigynous germination, remember the cotyledons are carried above the soil due to the elongation of the hypocotyl. So you have option B. I usually remember this by just saying, by using this keyword. So in epigynous germination, you have the hypocotyl growing fast, while in hypogynous germination, you have the epicotyl growing fast. Okay, so if you just have this code EHHE, so epigynous germination, the hypocotyl is growing faster. Then in hypogynous germination, the epicotyl is growing faster. So that will help you a lot to remember what happens. I mean, to be able to figure out the answer. Question number 19. Germination which results in cotyledons being brought above the ground is called dash. So remember what we just mentioned. So the right answer to this is epigynous. So in epigynous germination, the cotyledons are carried above the ground. So epigynous, cotyledons above the ground here. In hypogynous, from the name hypo, the cotyledons are below the ground. Question number 20. Hypogynous germination is characterized by, it's still repeated question now. Hypogynous germination is characterized by, let's look at the code again. In hypogynous germination, you have epicotyl growing faster and you have the cotyledons below the ground. So let's look at which of the options fits that description. D, elongation of the epicotyl. Question number 21. Epigynous germination of a seed is characterized by, it's the same thing. The question keeps, whenever they ask this question, it's usually repeated in a similar way. All right. because there are just two types of germination. There is no way you want to frame the question. If you can understand this very well, that a PG germination, hypoculture grows faster, and dicotyledon is above the ground. I mean, the cotyledon is above the ground. In hypogeal germination, a PG grows faster. Sorry, a epiculture grows faster, and the cotyledon is below the ground. There is no way you won't be able to interpret the question. Question 21 again. It says in epigeal germination, what happens? More rapid elongation of the hypoculture. No wasting of time. The right answer here is option B. If you consider question number 22, in a bean seed, absorption of water at the beginning of germination is through what part leads to the absorption of water? Now we need to look at, do I have the image of it? Let me see if I have that diagram here. Okay. Sorry, I don't have it. I wanted to see if I have the diagram of the seed. So basically, there is a part of the seed called the micropyle. It's a dry hole through which water is absorbed during germination. If you look that up in the textbook, you'll find that the right answer to this question is B. Then we'll move on to look at conditions necessary for germination. Question number 23. Remember the condition necessary for germination in the notes, which is here. We mentioned that factors affecting growth. Okay, for germination, you need oxygen, you need warmth, you need moisture, and you need a viable seed. Now let's go back to this question. Number 23. Water is necessary for a germinating seed because what does water do? Water helps to activate enzymes. It provides a medium for metabolic reaction, and it transports nutrients to the growing region of the embryo. So, which of that best fits into the option? Question number 23 would be B, activation of enzymes. Question 24. The two most critical factors required for the growth of plants are the two most critical factors. You need nutrients, you need water content of the soil, you need light intensity, you need topography, you need the type of plant hormone and enzyme, temperature, and pH of the surrounding. I think the answer is quite straightforward in this question. So for growth to occur, you'll be looking at intrinsic factors such as growth hormone and also enzymes, which are critical for plant growth. So two most critical factors required for growth of plant are you need the type of plant, the plant types of plant hormones and also the enzymes. This answer seems so plausible, but again, I might be considering option A, which also, a very, which is also very plausible. Water content of the soil, but in this case, no. I won't. Reason why A will not work is because growth can occur without even putting the seed in the soil. So that nullifies option A. Growth can occur without light intensity and without topography. Growth can occur um, over a range of temperature. So you see why it's good to logically reason out the answers. The options. So again, now this reaffirmed my resolve on option C, and that will be type of plant hormones and enzymes. The reason is that for growth to occur, there are intrinsic factors that are very important. And number one is the growth hormone, particularly in plants, which is auxin. Without that, growth is not going to happen. Auxin is an important hormone. Then you have gibberellins. You also have enzymes, which are critical for growth to occur. So let's move on to question 25. The most critical conditions necessary for seed germination. The most critical necessary for seed germination. Of course, water is important. So any option with water would have to stay. So we have three options with water. So we cancel out option D, right? What else do we need for growth to occur? Temperature is important. Oxygen is important. Temperature is important. Oxygen. And so anywhere you have oxygen, let's mark that as correct. So anywhere there's no oxygen now, we cancel it out. So I cancel out B. So I'm now left with what? A and C. Now let's go. Water, temperature, oxygen, as against oxygen, water, and carbon dioxide. So what do you think? Carbon dioxide is not critical for growth to occur. As a matter of fact, carbon dioxide is giving off in photosynthesis. I mean, oxygen is giving off. All right. So carbon dioxide is not important for germination to occur, as against um, oxygen. So I will. The right answer to this question would be option A. Next question, question 26. A germinating seed requires oxygen, which is essential for converting carbohydrates into glucose, transporting energy from one part of the plant to another, production of energy by oxidizing essential carbohydrate, hydrolysis of protein, formation of water molecules within the germinating seed. The answer to that would be for tissue respiration. Oxygen is needed for tissue respiration, and that leads to the production of energy. So oxygen will be needed to produce energy, and the right answer to that question would be C, the production of energy by oxidizing. Oxygen is needed to oxidize essential carbohydrates. Question 27. Water is required for seed germination to take place because now this is like a repeated question or in a different way though. Because here it talks about the importance of oxygen now, it's talking about the importance of water, but we have come across why water is necessary before, right? Okay, so these are ways that JAMB continue to repeat question. So question number 27. Want to take a guess? Activation of enzyme. And that was the same answer when the question was asked here. All right. Activation of enzyme in 2006. Okay, the question was first asked in 1980. And I'm sure it's been asked after 2006, several other times after that. Okay. So activation of enzyme is the reason why water is required for germination of seed. Okay, question number 28. Question number 28. Alkaline pyrogalore was used in an experiment. Then the experiment must have been connected with what? So if you remember, I told you there are experiments under growth. If you look at the book, there is a part that talks about experiment on growth, right? You have experiment one to demonstrate the condition necessary for germination. You have experiment two to determine the area of fastest growth in the root. Experiment three that demonstrate the root pressure um, root pressure using manometer. Okay, now let's go back to the question. It says alkaline pyrogalore law was used in an experiment. The experiment must have been connected with which of these processes? Question 28. So when alkaline pyrogalore is used in an experiment, it's not normally to show that oxygen is necessary for germination. So what alkaline pyrogalore does is that it absorbs oxygen. If you put alkaline pyrogalore in a controlled space, oxygen will be absorbed from that space. So the space will be devoid of oxygen. So and when you do that, you can now see the effect of oxygen. So if you do that in any experiment, carefully set up, oxygen will be, you are sure that that space will be devoid of oxygen. Then you can be able to tell what you want to tell with the effect of oxygen. So that experiment can be used to show that oxygen is needed for germination. And that would be option B. All right. Let's consider two more questions so we can have a total of 30 questions in this segment. And in the next video, I'll be moving to the next topic. So question 29. Growth can be determined in a population of Spirogyra by measuring which of these factors will you measure to determine growth in a Spirogyra? Remember, we said the most important measurement to determine growth, or the most accurate, is to measure the dry weight of the organism. So this is still a repeated question in a different form. So dry weight of the organism gives you a better description or a better understanding of the growth rate because you're keeping the cells constant by taking out the water. So you then want to see um, what has happened to the cell as it increased in size or as it increased in volume by taking out the water. Now, finally, question 30. Same question repeated over and over again. Water is required for seed germination to take place because it is needed for activation of enzyme. You can't miss this question again. Okay, so activate enzymes. So that is where we are going to stop in this section. If you think the video was useful, please like, subscribe, and share. And then I will see you in the next video. And also don't forget to leave a comment if you think that there is any answer that is not so clear, or you think you disagree with my choice of option, then you could also give your own explanation. I mean, it will be useful for others to have that debate and then learn from you as well. So there are other questions in the book. If you find the book Explicit Biology, there are also the other questions and the explanations are at the back of the book. In the next video, we'll be looking at coordination and control, which will be topic 15. So I sign out here. I'll see you in the next video. Bye for now.